access to the exam is through you.
Yes. Yes.
Okay.
Okay. So today we're going to start with the first topic that will be in your second exam, which is methods in protein biochemistry. We're going to cover several techniques and methods to study proteins in general, either for purification of proteins or characterization or other techniques. Some of them you will see. For all of them, you should know what is the principle of each techniques. For some of them, we're going to cover very in detail some things that are essential for that method. And for others will be the overall, what is the principle and the application of the techniques. Okay. But before I start with the new topic, as I was mentioned, if someone wants to know exactly, look at the exam, just contact me and we can, during office hours, look at the exam. In general, it was pretty well. You guys did, the average was 80, which is good. There were several technical issues, so I will ask you, please, next time, perhaps I know it's technological issues, but please have a plan B, etc., right? Because I can help until certain point, but other than that, there is not much that I can help with. So please plan accordingly, follow some of the tips from IT, etc. Okay? I will prefer to have the exam for 100% to be in person, but it's not possible now. Yes. Someone has a question?
Yes, it's me again. Sorry.
Yeah.
So yeah, I think I noticed your episodes are usually like around 11-ish. I'm usually at work at the time, and like usually I'm off by the weekends. Is there like, would there be like any other time that you'd be willing to meet, or should I email you about that?
Yes, please email me for that personal issue. Conflict with the schedule. Just email me. Okay.
Okay.
Okay. So before I'm not going to cover the whole exam, but some certain questions that some of you got incorrect. I just want to clarify. For example, this one, it was pretty straightforward, right? So true or false, saying living organisms are in equilibrium to maintain homeostasis. Recall that living organisms are not in equilibrium. Recall that equilibrium is not change. Right, there's no change in the substrate and the product, which is impossible in an organism, right? Homeostasis, yes, we have homeostasis, and that's basically the ideal state in a living organism, right? So here the answer was false because we are not in equilibrium, because we're talking about living organisms. Here, there were two questions related to this oligopeptide. So by now, you should know right away without any problem, right? That we have 1, 2, 3, and 4 amino acids, right? You should recognize right immediately at this point that this is the peptide bond. So one of the questions was, how many peptide bonds do we have here? So if we have 1, 2, 3, and 4, we should have 3 peptide bonds, which is here, right? 1, 2, and 3. And obviously the sequence of amino acid, well, you have to recognize right away, right? We have leucine right here, cysteine, which is one of the 2 amino acids that have the sulfur group. We have here tyrosine and glutamic acid, okay? And the problem for isoelectric point, I think the majority got correct, which was good. This is another question that I saw many of you got incorrect. It was just mentioned a protein, doesn't matter the name. It's a protein that it's in the lipid bilayer, right? So recall that the lipid bilayer is nonpolar, right? Therefore, and then it's blank and say amino acids are likely to be found on the interior of the surface. So on the exterior, because we're talking about non-polar environment, will be amino acids that are non-polar. But in the interior, we're talking about hydrophilic amino acids. Okay. Someone has a question? Any questions? Someone was raising the hand. Go ahead. You have a question?
I think Bakari still just has his hand up from
Oh, sorry. So that was from before.
Good.
I forgot to lower my hand, but I think maybe just if I could make a side comment. I think probably what threw some people off. I can't speak for people, but maybe what would throw a person off when looking at this and the way the question is formed is possibly, I guess, when we think about phospholipid bilayer, we're thinking about, okay, like the head would be pointing towards water, and the tails would be facing away from water, I guess. So I guess they thought that some people may have thought that that would mean the answer would be hydrophobic, in which they were thinking just about the tails rather than the entire structure.
Yeah, well, you say interior, so yeah, maybe because they thought it was exterior, but yeah.
Yeah.
Okay.
Professor?
Yes.
I'm just a little confused because I remember reading, and if you could just go over this a little bit, because I remember reading that the amino acids face towards the interior because they don't want to be around the water or the outside of the cell, like the solvent that's outside of the cell. They face inward because they're hydrophobic and they can't react. I don't know. Can you just explain? Because I remember reading that. I don't know if it's wrong or right.
No, but here it's saying that there's a lot of proteins that are in the lipid membrane. These red little things are proteins that are that the case. If the question will say outside, so outside the environment in the interior of the lipid membrane will be non-polar, and the answer will be hydrophobic. But the question is inside the protein, right? So inside will be hydrophilic because they're not going to interact with non-polar. They don't want to interact with non-polar environments. So recall that in the middle, if the protein is here, this is a non-polar environment. So outside, yes, you're going to have amino acids that like to interact with non-polar. Those are hydrophobic, right? In the exterior of the proteins outside, but inside, all the hydrophilic, the one that they don't want to interact with non-polar environments are going to be inside. So that's basically the answer. Okay.
Professor, if it's inside and you said it doesn't want to be around water, then why is it hydrophilic? Wouldn't it be hydrophobic?
In the interior, they're gonna be, so in the exterior will be hydrophobic, and the interior will be hydrophilic.
And it's hydrophilic because it doesn't want to be around water?
Hydrophilic doesn't want to be, in the non-polar. Remember, we're talking about the membrane. In the membrane, it's non-polar. There's no water, right? So in the membrane, remember, we're talking about the lipid bilayer is an environment that is non-polar. It's not polar. Okay.
Professor, do you mind if I try explaining it to her?
Yeah, sure. Go ahead.
Okay. The way that I think about it is that the inside of the lipid membrane is all the tails of the phospholipids, right? And that is a non-polar environment. So when a protein is inside with those tails, it'll fold so that all of the non-polar side chains will be facing outside, and it'll put all the polar side chains on the inside, so folded in. So the question is asking what's the interior facing helical surface. So basically what side chains will be all joined together on the inside of the protein inside the membrane, if that makes any sense.
Yeah. Maybe it was a wording, but basically just have to think about where is the protein, because normally we will think, okay, if it's in the cytoplasm, inside, what's going to happen inside as Nicole, right, will be in the protein, will be hydrophobic. But here we're talking about the membrane, which is a different environment. So depends on where the protein is where the amino acids are going to be interacting.
Professor, so in terms of the membrane would be hydrophobic. Is that referring to the tails?
Can you repeat again? It's hydrophobic, correct. So hydrophobic, you're going to have all the amino acids that like non-polar environment on the outside of your protein, right?
Okay, thank you.
Okay, so then this one was really, really easy, but I don't know why some of you got a little bit confused. It was not even a calculation, but very straightforward. It was telling you two reactions, and recall that the free energy, right, when it's negative, it's a spontaneous reaction, and always, or the majority of the reactions in the body, it will couple with another reaction that is spontaneous, right? Which will give the delta G a negative value, and therefore it can occur and can be spontaneous. So basically here it was just the difference, and the answer was -8 and it's spontaneous. Okay.
Professor.
Yeah.
Regarding that question, because we have some similar questions like that on the progress assignments, and the answer was like non-spontaneous. So this is why it's kind of confusing.
If you want, we can talk about that. I don't recall that, but we can look at that, okay?
Okay, all right, okay, no problem.
Okay. Well, basically, that's it. That's got my attention. The rest was pretty straightforward. And that's it. Any other questions? Recall that the exams will be always during the time, during the class time. So it's from 6 to 7:45. Some of you, I have to stop the exam because it goes longer. So just plan accordingly. But some of you finish the exam by 30 minutes, you were done, or 40. So, okay. Any other questions or concern or comments from the exam? You think was fair? Mm-hmm.
Professor, will you post our the questions that we did? Because I see right now it's still closed.
Yeah, I mentioned that if you want to see your exam, you have to make an appointment with me, and then we can go over the exam. Okay. So here are the learning objectives for the first chapter for the second exam. As I mentioned, we're going to cover several techniques to study protein in general. So first, we mentioned this in the previous chapter, right? There's amino acids that are uncommon, and these amino acids, so the 20 amino acids that you already know are the 20 amino acids that are made in the ribosomes. There's some modification that occur post-translational, meaning after the protein is made. And some of very common modification in the amino acid are phosphorylation. So phosphorylation occur in the OH or several amino acids. And this is a very important type of modification. Certain enzymes can be activated or deactivated or inactivated with the phosphorylation itself. The enzymes that are in charge of doing or passing the phosphate from ATP are kinases. They're called kinases. We're going to cover enzyme in this for this exam in chapter 7, but I have mentioned this. So this is a very common type of enzyme that is called kinases that add the phosphate to that amino acid. And this is from the previous presentation. The case of serine and threonine, the OH on the R group, it's the one that actually the kinases here transfer the phosphate group. There are more that you will find, but again, like the example of collagen, the hydroxyproline, the OH group, again in the R group of proline, this is important for the structure of collagen. And there are others modification, but the important part to know is that this happens after the post-translational modification. So after the amino acids are made, okay? There are other proteins that are covalent bound to a non-protein entity, meaning that are not proteins. For example, lipoproteins, it's a protein that is linked to a lipid molecule. Glycoproteins, that is linked to a carbohydrate, etc., right? Phosphoprotein, that is linked to a phosphate. So all these modifications are important for many type of proteins occur after, right? After the amino acid is made, okay? Okay, so let's start with all the techniques actually to study proteins. So proteomics, when we're talking about proteomics, and in the first presentation, we talk about the omics term, that means basically the set of, in this case, we're talking about proteomics, meaning it's a set of proteins that is expressed in certain circumstances, right? So imagine if you have a proteomic analysis from a cell that, from a patient that has Alzheimer versus a healthy patient. So that is, if you look at the proteomics, that means that you're looking at all the proteins that are occurring, for example, in Alzheimer disease versus a healthy donor. So it's all the proteins that are occurring in that specific circumstances. Here we have genomics, right? Omics, all the genes. Proteomics, all the proteins itself. So when you hear like, oh, proteomic analysis, well, there's certain techniques that you need in order to do the proteomic analysis. So for now, just have in mind, right, that proteomics means the entire proteins of something from, for example, the example of the cells from Alzheimer patient, okay? Under that circumstances.
So it's referring to the proteins in I guess the cells of reference, I guess.
Can you repeat that?
Like proteomics, so it's just looking at, I guess, specific cells, not like in the whole body, but just like, I guess, whatever you're looking at specifically.
Yeah, whatever you're looking at. So the entire proteins of, I mean, it can be the whole body, right? The whole organism, the whole, I don't know, yeah, whatever you're looking, that it's proteomics means proteins. Omics is the whole, the whole set of proteins. If you're talking about genomics, is the whole genes in that. Transcriptomics, we're talking about the transcript, the mRNA, okay?
Okay.
So when we're studying proteins, actually, you need to isolate the protein that you're looking for, right? That is the purification. You need to get rid of a lot of things that you're looking for a specific protein. So you have to get rid of everything that is not your protein. So you need certain technique to actually remove everything that you don't need, right? We know now, right, that amino acid, which are the building blocks of proteins, are different, depends on the chemistry of that amino acid. It can be charged, polar, non-polar, etc. Right? So it depends if you're studying a protein that is polar or non-polar, positive, etc. Then you will find the technique that you will need. Okay. Obviously, the protein is not itself, it's the whole cell and then you will have 1,000 and 1,000 of proteins. So you need to find methods, right? And it's just one method, it's several methods in order to get the proteins that you're looking for. So that's why they fish them out from the complex of a cell, for example, okay? This is a luciferase. This is basically what certain organisms are bioluminescence because they can actually emit light. And this is because of a reaction that emits lights. But the point for this slide is basically, imagine that you're looking for luciferase for that specific enzyme. That enzyme is in an organism. So imagine that you want to isolate luciferase from this insect. What you have to do? Well, it's the whole organism, right? You have to isolate that. So here, or also, what you have to do is get rid of everything that you don't need. So you will be doing a bunch of methods, for example, centrifugation. You have to lyse the cells to remove everything that you don't need. And then you will have different different fractions, right? So, and not all of them will have the luciferase. This is easy to look at the protein of this specific enzyme because we know it emits light. So it's easy for me, for example, if I lyse the organism and I collect all the tubes, you will have a bunch of tubes. You're not going to be able to see, but then you perhaps you will be able, because it's luciferase, you will be able to read the luciferase itself. So from all these tubes that we're saying is that the number 2 is where your enzyme is, you will find. So the bottom, the point here is that you should know the protein that you're studying, because in this case you will say, well, I'm looking for luciferase. I know it emits light, therefore there is a method to look at the light, right? If you don't know anything, that will be a little bit complicated. This is another example. If you are looking for the enzyme, which is lactate dehydrogenase, a method to study lactate dehydrogenase, which is one of the steps after glycolysis, you have lactate and then pyruvate. Perhaps you cannot quantify pyruvate, but turns out that this enzyme, what happened is the lactate dehydrogenase emit, right? One of the products is NADH, and NADH will absorb light at 340. So therefore, you have your tube, you put the enzyme, and then you look where in the tube you will have the absorbance, right, of 340. So you will need a method to know where, in order to know where is your enzyme, okay? So imagine that you are studying certain reaction and you will say, where is this, if the reaction occur or not. One of the ways through absorbance, you look at the absorbance at 340, okay? So one of the first steps in purification is actually understanding the function of the protein that you're looking for, okay? So as I mentioned, you have to remove everything in order to purify, everything that you don't need, right? To purify the protein that you're looking for, the cells, the tissue, etc. So it's very common, right? When you go to any lab, you will find a centrifuge. And this is a centrifuge right here. For example, if I'm looking in the liver for certain enzyme or proteins, how am I supposed to study the protein? Well, I have to lyse, literally break apart the liver, in this case, by any method, either sonication or detergents that you break apart all the cells, and then you will have the extract. But that's not it. That's one of the first steps. Then you have to collect that, and then you have to centrifuge to different forces, right? Depends of the molecular weight of whatever you're looking for, you're gonna separate. So it's important to know, where is the protein that I'm looking for? Is it in the cell membrane? Is it in the cytoplasm? Where is it? So depends on what you're looking for is the centrifugation that you have to do. Okay, for example, here, after you get the tissue from the liver, right, either with enzymes or literally force, like sonication, etc., then you're gonna do several centrifugation. You don't need to know exactly these numbers. I'm not gonna ask, but yeah, you should know that different centrifugation will give you different components of the cells, right? At the beginning, for example, you will have the whole cell, and then the plasma, the nucleus, the mitochondria, etc. And then again, you should know where is your protein in order to study the protein that you're looking for. So at the beginning, you have the whole extract, and then you centrifuge, you will have the nuclear fraction, and then the mitochondria, and then the membrane and the cytosol. The majority or many of the proteins that in my lab, for example, we study is in the cytosol, but there are other proteins, right? It can be in the mitochondria. So then you should know where is it in order to know the centrifugation that you need to use, okay? Obviously, any questions for now? No. Okay. So then obviously every time that you try to purify the protein, you will be concentrating, right? And you're gonna have more activity in the protein itself. So you have here, for example, the cell extract. Here you have units. This is just arbitrary units for whatever the protein is. The protein, the function of the protein. So let's say you have 20,000 activity units, and at the beginning you have 2,000 milligrams of proteins at the beginning here. So then you centrifuge, and after all this centrifugation, turns out that the protein that you're looking for is in the cytosol. You can see here that the protein is, the milligrams are less, it's 500. So therefore, in this case, the specific activity, which is basically the activity units divided by the total protein, will give you the specific activity. In this case, you have 10 units per milligram of protein, and in this other case, you have 36. Okay, so you're going to be purifying in all the steps that you have to do. And here's basically how they explain. So you have your protein. Imagine that you're looking for this protein that is represent the red little balls right here, or marbles. Here you have activity, but obviously you have other proteins, right? It's not pure. It's not. You have other things that you don't need, and it's holding, right? Basically the specific activity of your protein. When you get rid of all the rest that you don't need, you will have higher specific activity because your protein will be more, you will be doing the purification of the protein that you're looking for. So you have the protein, you have more higher specific activity of that protein. So if we go back to the example of the enzyme that emit light, luciferase, then you will be able to actually look the luciferase because you get rid of everything that you don't need. So that's basically the specific activity. The total amount of activity of the target divided by the total amount of protein in the fractions. Mm-hmm. Yeah, go ahead. There's a question.
So in order to get the specific activity, you have to first do the centrifuge thing?
Well, this is one of the examples, yes. A very basic, remember we're talking about several methods, but one of the basic methods is first get rid of many of the things that you don't need. One of them is actually lyse the cell and then do a lot of centrifugation. Mm-hmm. And that's not it. You have to do more. I'm going to explain a little bit more. But that's basically so you can visualize what is actually a specific activity. Okay? Okay, so yeah, when you do the centrifugation, each fraction contains less total protein, right? Because we're getting rid of many of the proteins we don't need and a lot of organelles and a lot of things that we don't need. But the specific activity, it's increasing, okay? Let's see here. Okay. I'm gonna go back to the specific activity in a moment. Okay. As I mentioned, there is no one single method when you're studying proteins at all. This is just to start. For example, in my case, I do cancer. So one of the things after I treat my cells, what I do is, I lyse the cells. I remove everything. I break apart the cells, and then I look for specific proteins. I extract the protein from the cells, and then this is the first step. After that, what I have to do is a Western blot analysis. I'm gonna explain Western blot in this class, but basically is to look at the expression of different proteins. But doing the centrifugation, the lyser, etc., is just the beginning of whatever you're looking for. So after you're done with the centrifugation, one of the methods is salting out, and this is basically to precipitate a protein. Basically, the principle, you can see here, solubility versus salt. It's that at the beginning, when you put certain amount of salt, sodium chloride or potassium chloride, any salt, at the beginning, you have a very good solubility because it's a good interaction between the salt that you're adding, the water, and the protein. But there is a point that you add an excessive amount of salt, and that's where you can see here the solubility decreases. Therefore, because the salt will compete with the water and the proteins for the interaction, therefore, the salts that you can see here and all the proteins will interact, it will precipitate. So you can see here in this cartoon is basically what it's saying is that with a excessive of salting out, that means that the salt is not going to be soluble anymore with the water and the protein, but the proteins are going to look for each other and get together, and then we'll get aggregate of that protein, and then it's going to be you're going to have the proteins all together. Okay, so you can have excess of salt, as you can see here. If you increase the concentration, then the solubility will be less. Okay, and that's how basically how you can get some of the proteins, right, that you want. This is one of the techniques. Then how you get rid of all the salt? Well, you can do dialysis, right? So basically, if you put that in a back of dialysis, all the salt will be out and the protein you're gonna be inside, okay? That's one of the things, one of the methods. Okay. So another methods are to study proteins and make purification are actually chromatography. There are different type of chromatographies. And basically it's some beads or some beads that are in a column, and those beads can be either, let me see, you can have either gel filtration, okay? I'm gonna go by one. Size exclusion or gel filtration, meaning that how you're gonna separate in that chromatography is based on the size of the proteins. And in this case, the bigger proteins here are gonna travel and the small proteins are gonna go inside the beads, the little beads. It's like little holes, as you can see here. So that is based, gel filtration or size exclusion is based on the size. The other type of chromatography is ionic exchange. Basically, they're beads that are a positive charge or negative charge, and then you're gonna separate based on the charge or affinity. I will explain one by one, okay? That's why I'm saying that there is no a single method, and it depends on what you're studying. Gel filtration, separate based on size. So for example, you add the whole imagine we have the whole lysate, but you have to separate. So the small proteins are gonna go through the beads inside the column a little bit, and the big proteins are gonna go directly, gonna pass directly to the column, right? So larger proteins would migrate faster than the smaller ones, okay? So imagine you collect all of the tubes and you will say, well, the molecular weight of my protein is this, therefore will be perhaps in tube number 3, 4, 5, okay? But then you separate based on the size, but then you need another method to further analyze your protein. But this is the principle for gel filtration chromatography. Ion exchange chromatography, these beads that I mentioned in this type of chromatography are charged. Therefore, if they're positive charge, right, they will interact with the negative charge protein and vice versa. The negative charge beads are gonna interact with the proteins that are charged positive, okay? So two matrices we have positive charge will be for an anion exchange chromatography. So they want to interact with negative charge, and the negative will interact with the positive charge, okay? Again, you need to know what is the characteristic of the protein that you are studying in order to say, well, I'm gonna use a positive charge type of chromatography or negative charge, okay? So here represents the negative charge beads. Therefore, the positive charge proteins are going to be happy, right, to interact. The negative, by the opposite, are going to elute first. They're going to pass to the column really, really fast. Okay? And then the way to elute, because, okay, you trap, in this case, the positive size, positive protein, sorry. What you need is actually a solvent that is either that compete with the protein. In this case, the protein is positive and the beads are negative. Well, you need a positive a solvent, right, to compete. Therefore, the proteins that are attached to the beads can elute. Okay, then that's how you have the exchange, right? And then you have your proteins. Any questions? It's clear? Elution is how we call when you remove and you get the proteins from the beads. Okay. So affinity chromatography, as the name implies, those are type of chromatography that the beads are covalent linked to something that the protein has affinity with. For example, if a protein, the protein or that I mentioned has a ATP or a phosphate group, what you can do is those beads can be attached to ATP groups. So when I add that specific enzyme or whatever that like the phosphate, then will bind. That's the affinity part. Okay, that's one of the examples. Okay. Questions?
Could you repeat that, please?
The affinity chromatography are beads, right, that contain a covalent link, a molecule that is covalent linked to the beads that are with affinity of the protein. For example, if a protein is like a phosphate, you can add ATP to the beads, a covalent linked ATP. Therefore, when you add your mix of the protein, it will bind to that ATP. So it binds to the beads that contain the ATP, and then you get rid of all the proteins that are not they don't have affinity for ATP. Okay, so that beads will have something that is covalent, that are covalently bound to something that the protein is have affinity to. Okay, so that's
Professor. I'm sorry. Do you mind going back to the one where you use the buffer? I'm just a little confused. How does the buffer help separate from positive and the negative?
So are you clear with this first part?
Yes.
Okay, so imagine you put the mix, right? So all the positive proteins are going to bind to the beads. How are you going to get, how then, okay, they're bound to the beads, and then how are you going to get the proteins from the beads? You need something, you need a buffer, you need something to then deattach the protein that you want, because that's what you want, right? You want to study the proteins. So your proteins, you put the whole mix. All the proteins that are negative are eluted, meaning you will have, imagine all the tubes, they're drops, right? You already get rid of all the proteins that are negative, but the proteins are attached to the beads. So you need something, a buffer that compete. Now that are the proteins that you are looking for. Those proteins are positive. So you need a buffer that is a excess of positive charge. Therefore, when you have a positive charge, it's going to bound to the beads, and the proteins, it will be compete basically, and it will exchange, and therefore you will have all the proteins that are positive charge. So you need something in this case that compete with the proteins that you want in order to remove it from the beads, because you need the proteins, right, to analyze. Okay?
Thank you so much. That makes sense.
Okay, good. And the same for this example that I mentioned with ATP. Okay, fine. I put ATP on my beads. I put on my sample, and all the proteins that are they have affinity with ATP will bind to the beads. How am I gonna elute the proteins from the beads? What do you think?
Can you use a buffer again?
What buffer? Or what are you gonna use?
Something that would also bind with ATP?
Correct. Something that will bind to the beads, in this case ATP, therefore it will be stronger, right, than the proteins. You get rid, then your proteins are gonna deattach from the beads, and then you're gonna collect the proteins. That's it. That's basically the principle of that. Okay, that's affinity chromatography. Let's see. So you have the beads, the target protein, covalent bound, and then etc. Okay. ATP. Yeah, ATP. So I'm gonna put an animation so we can, wrap this part of chromatography, and then we're gonna continue. Let me stop here. Okay. Wait. Share.
Column chromatography. Column chromatography is a method of protein purification. A protein mixture is added to a column containing a solid matrix. As the mixture flows through, proteins are separated based on their different chemical or physical interactions with the column matrix. Column fractions are then tested for target protein activity, and those fractions containing the highest activity are combined for later use. There are three major methods of column chromatography: gel filtration, ion exchange, and affinity chromatography. The first method, gel filtration, or size exclusion chromatography, uses porous beads for the solid column matrix. The pores in the beads are large enough to allow for small proteins to pass through, but exclude large proteins, which flow around the beads. As a result, large proteins flow through the column much faster than small proteins, which take longer, slower paths through the numerous paths created by the pores. The second method, ion exchange chromatography, uses a charged column matrix to separate proteins based on their overall positive or negative charge. Note that while a negatively charged matrix is shown here for cation exchange chromatography, proteins can also be purified using a positively charged matrix for anion exchange chromatography. For example, a positively charged protein is attracted to negatively charged beads, whereas negatively charged proteins are repelled by the negatively charged beads, and uncharged proteins flow through the column matrix unimpeded. As a result, the positively charged proteins will move slowly, while the negatively charged and uncharged proteins will flow through the column much more quickly. After the negatively charged and uncharged proteins have flowed through, an elution buffer with sodium chloride containing positively charged sodium ions is added to the column, displacing the positively charged proteins from the beads and helping to elute them from the column. The third method, affinity chromatography, is more specific than the other two methods. A ligand with a high affinity for the target protein is covalently bound to the gel matrix. The target protein will specifically bind the matrix, while other non-specific proteins flow through. As a result, the non-specific proteins will move through the column much more quickly. To release the target protein, an elution buffer containing an excess amount of a competing ligand is added. Target proteins can also be released by elution buffers containing an altered pH or salt concentration. The competing ligand binds to the proteins, displacing them from the matrix. In summary, there are three major methods of column chromatography that can be used depending on the properties of the proteins that need to be separated: size for gel filtration, overall charge for ion exchange, and the availability of a high affinity ligand for affinity chromatography.
Okay. So we are clear with chromatography, right? Okay. Yes, okay, good. Okay, so the other type of chromatography that is very common, it's HPLC, which stands for high performance liquid chromatography. Some people say high pressure liquid chromatography, but because it uses pressure, but in reality, or the correct way, the correct definition is high performance liquid chromatography. And basically, no matter what, because some of them are reverse HPLC or just HPLC, you will have, the principle is that you have to have a solvent right here and a column. We have columns again. But here, it's a column that is packed. Either it can be, for example, one of the common is C18, which is a non-polar column. Then the solvent will be polar. So if you have non-polar pack, what's going to happen is that the non-polar molecules or proteins, in this case that you're separating, will interact and stay longer than the proteins that are polar, right? So if this is non-polar, the polar proteins are going to elute, right? Not elute, like pass through the HPLC. And here, this is a chromatography graph. You will have a time and the absorbance. So if you have a column that is non-polar, the molecules or the proteins that are polar are going to pass first because they're not going to be interacting, right? So if it's not interacting, you will have certain peaks, and certain peaks will be equivalent to certain quantification of that specific amino acid or proteins. Okay. But basically, it's a column matrix, and then it will be based on the affinity to the column or not. Okay. Okay. So I mentioned that I was about, we will see the specific activity again. This is a table showing that, for example, what is the specific activity of different, when you start with cell extract and then you use different methods of chromatography. We can see that the extra, you have a lot of protein. Obviously, you have a lot of a mix of different proteins. you have 2000 milligrams of a specific protein. And then when you are doing the centrifugation, fractionation, you will have less, obviously. When you, if you do a salting out, then you have more concentration of the protein that you're looking for, but you have less total protein in general, right? It makes sense. And then let's say you use gel filtration chromatography, ionic exchange, or affinity. So you can see that the total protein itself will decrease, but the specific activity, right? The activity of the protein is you are looking for will increase because you are concentrating basically, and you are doing a purification of the protein that you're looking for. So it makes sense, all these numbers. Okay. And if you actually run the sample, you are in the lab and you say, well, I'm gonna get my enzyme from this liver and I'm gonna do lysate, I'm gonna get rid and break apart the cells. Then if you run this in a gel, you can see here a lot of bands. Each one represents a specific protein. So obviously in the beginning you have a bunch of bands because you have a lot of proteins. When you start doing all the purification, all the steps, you will see less and less band, which means you are purifying the protein that you're looking for. And in this case, for example, affinity chromatography was necessary in order to get your single band. And this is the protein that okay. And questions are about here? I think it's pretty clear, right? Okay. And this is where we have another method, which is gel electrophoresis. This is basically a gel, right? Where you can actually separate the proteins. You can use basically gel electrophoresis. You will use in this case prolacrylamide gel is basically like a jello where you can separate proteins. Remember when we were studying nucleic acid, how to separate DNA, RNA, you use agarose gel. And this is just because the size of that specific gel in nucleic acid are bigger than protein. So it depends on the size. In this case, for protein, we use prolacrylamide gel electrophoresis. And it separates the size, the proteins based on the size and the charge, okay? And it's important, you can use different percentage of the polyacrylamide, and that will make a difference on the size, right? And the pore. So if you have a very high molecular weight of proteins, let's say to 400 kilodaltons of protein, which is pretty high, you will need low percentage of the prolacrylamide gel. So this cartoon represents the polyacrylamide gel electrophoresis. You will need electrical field, as you can see here. When you add the proteins right here, the smaller proteins are gonna be down. You will find the proteins, the low molecular weight in the lower part, and the larger protein will be in the top, right? If something that is common to use in order to give negative charge, a uniform charge of a protein is use the SDS, or sodium dodecyl sulfate. The SDS is a detergent that give negative charge of all the proteins in general. So therefore, when you are doing this polyacrylamide gel electrophoresis, what are you separating is based on charge and not based on, based on size, not based on charge. Because all of them, SDS, what it does is give negative charge of the protein. Therefore, whatever you are separating, if it has the SDS, will be, you will separate based on the size of the protein, okay? When you add the SDS, oh, yeah.
If you use the SDS, you can't like use the protein, right? Because it's a detergent.
Yeah, correct. Yeah,
Okay.
very good. So when you add SDS, yes, correct. You are, and it's here. When you add SDS as a detergent, you denature your protein. Your protein is not native anymore. It's not functional anymore. You're correct. When you add that, and we add the SDS and then we boil it. We add temperature. Therefore, it's not functional anymore. I mean, it's functional just to look at the expressions in the gel, but you cannot use it anymore. Mm-hmm. So yeah, you're correct. When we run a page gel, this polyacrylamide gel electrophoresis, we say page. When we run a page gel and we add the SDS, we call SDS page, okay? And you should know that the SDS will create this detergent, yes, as you will say, it denatures the protein, you cannot use the protein, give a negative charge to the protein, and it's like the DNA, right, and RNA that are negative charge. Because they're negative charge, obviously they're gonna go to the positive sides, and you're gonna separate based on the size, okay? With the larger proteins on the top, smaller on the bottom, okay? Yeah, so you add SDS plus the high temperatures. Okay, so here I just want to make sure you guys, we are in the same page. And so if you can say true or false, if native proteins, we're talking about proteins which are in their folded and assembling form and are fully functional. Let me put the question. So, true, false. When I'm saying native proteins. Okay.
Professor, I don't see the poll.
Oh, I just stopped the poll. You can chat me if it's true or false. If native, when we're talking about native proteins, that means are folded and fully functional. Okay. So yeah, 89% say it's true, which is correct, right? And those 4 students that say false, the answer was actually true, because we're talking about, remember when we studied folding the protein, right? The chaperones that are involved, some of them, they require the folding of protein where it's fully, where it's folded is native. We're talking about when it's fully functional. And when we add the SDS, we are denature the protein. They are not native anymore. So if you want to run actually a gel with native proteins, the only thing you have to do is just add it, right? You don't add any detergents, you don't heat your protein, because then you're gonna denature and you're gonna disrupt the secondary structure and etc. Okay. So that's the difference between denature and not denature gels. So it's important to know what is the difference, right? If you have a gel, right, you run a protein that is native and a protein in a native form, or is in a native form or denature form, you will have different patterns, but it's the same protein. It's just that it's denature. For example, here, if you run the protein you will have one band, but perhaps when you run the SDS page, you will have two bands in different molecular way because, for example, this protein that is A could be a heterodimer, right? Therefore, you will have two subunits of that enzyme, a protein. That's why you see two molecular weights, but two bands, but it's the same protein. Or it can be that you see this protein, B is another protein. And when you run the SDS page, turns out that you see the molecular weight, it's different, but you see a single band. And the only thing that happened is that this particular protein is a homodimer. That means that it's two identical units, okay? So the protein can be heterodimer, which is different, okay? Or it can be homodimer, okay? So under the SDS page, you will break the homodimer, in this case, into two subunits, right? And you will have different bands, okay? So it depends of if it's native or SDS page, and if it's heterodimer or homodimer. Questions? Good. Okay. So again, this is before the SDS is perfect, it's native, it's folded, it's functional, the protein. After you add SDS, after you hit the protein, then you have all of them are equally charged, negative charge, and it's easy to separate using the SDS page, right? Based on just the size. But if it's just the native gel without adding SDS, then you can separate the protein based on size and charge. Okay. Any questions? I'm going to put another video because sometimes it's better to visualize after we cover this, and then you can see the animation. At least for me, sometimes it's easier to look at the animation. It's easy to get the concept. Okay. So I'm going to share this. Hold on. Whoever had the book, you can go back and look at it. If you don't have the book on the homeworks, which the homework is available for chapter 5, you will find the videos if you want to go back and review some of them. Okay.
SDS gel electrophoresis. SDS polyacrylamide gel electrophoresis, or SDS PAGE, is a laboratory technique that can be used to determine the approximate molecular mass of a protein. First, the proteins are treated with sodium dodecyl sulfate, or SDS. SDS denatures the folded protein and disrupts the secondary structure. Negatively charged SDS molecules coat the protein chain and give it a significant overall negative charge. SDS coats all proteins evenly so that the negative charge is proportional to the size of the protein, and all proteins in solution have the same charge to mass ratio. Moreover, because the proteins are denatured and form random coils, protein shape is not a factor in SDS PAGE. To ensure that proteins in the SDS solution are fully denatured, the sample is heated in a boiling water bath. The sample is then pipetted into a well in a polyacrylamide gel matrix. An electric field is applied across the gel, which causes the negatively charged proteins to migrate across the matrix, attracted by the positively charged anode at the other side. While large proteins are restrained by the polyacrylamide gel, small proteins are able to move quickly. As a result, proteins become separated into bands within the gel according to size. The rate at which proteins move through the gel can be adjusted by changing the percentage of polyacrylamide in the gel. For example, increasing the percentage of polyacrylamide decreases the rate of protein migration, and the separation between fast-moving small proteins is improved at the expense of large protein separation. By measuring the observed migration distance of the unknown protein against that of known proteins, the molecular mass of the unknown protein can be determined. Finally, the protein bands are not visible until the gel is treated with a dye, such as Comasi Brilliant Blue, which selectively stains proteins. In this photograph of an SDS page gel stained with Comasi Blue, we can determine the molecular mass of our target protein by comparing it to a solution of marker proteins with known molecular masses. Here, our target protein is approximately 110 kilodaltons.
Okay. So that's basically SDS gel, and this is what I mentioned in the video, right? So again, all the proteins are going to be negative charged, therefore they're going to go to the positive side, and we're going to separate based on the size. Okay.
Professor,
Oh yes.
is that video in the slides or no?
The video, you have access to the book, you will find it. If you don't have
Okay.
access to the book, it will be available in the homework, which is available.
Okay. Thank you.
You're gonna review.
Okay, thank you.
And then you can see here, we always need a marker because you need to know where you have a reference, right? We call a molecular marker. In this case, you can see here, it's just a mix of different proteins. So you add the marker, you can see where it's 200 kilodaltons. Remember when we calculate the daltons, kilodaltons of the proteins, basically it's because you need to know what is the molecular weight of your protein. If I'm looking in this case, well, the target protein, I know it's around 120 kilodaltons. And I should know because if I don't know, that will be a little bit hard. So we always use a molecular mass, and as it was mentioned, right, we need something to stain the proteins, because when you run it, you need something, right, a color to look at the proteins. In this case, we use Comasi Blue, and it's blue, literally. You can see all the bands that are the protein that are blue, okay? So that's SDS page, and then how to visualize, and we use molecular mass, okay? Any questions? All right. So if you're clear with this, that this next question will be super easy for you. So if you can't either copy or copy in your notes, but try to solve this. It's like, congratulations, you have purified your protein X. You and your research partner have subject your protein to proacrylamide electrophoresis, which is basically what we cover. You treat the protein with SDS and mercaptanol, which is another detergent, and you boil it. You add heat, right? And your partner run a native gel without the SDS and without any reducing agent. Then you run the gel, and turns out that you observe one band on each gel, but your protein that you add the SDS would migrate at 23 daltons protein, while the partner, right, you both run a gel, but one is SDS page and the other is just page. So a protein around 92,000 daltons. So after you discuss that, you see the different patterns in the gel, you feel that the protein that you guys were looking in the lab, it's a dimer, tetramer, or monomer. And then the SDS and reducing agent have, and then you have to select. either degraded the protein, fold the protein to smaller components, denature the protein, or alter the ability of the protein to migrate through the acrylamide. Okay? So try to read it again, try to look at the gels and then the molecular weight, and then answer if this protein is a dimer, tetramer, or monomer, and what is the effect of the SDS and the mercaptan. If you have questions, let me know, but go ahead.
I'm sorry, is there a poll?
No. You done?
No, I was just like really scared because I didn't see a poll.
No, there's no. Actually, I want to put you guys in groups. This is a nice exercise to discuss. And again, I would prefer to be in the classroom because it's easier to see, the interaction between you guys. But anyway, so I'm gonna try again. If you copy this, I will do the polls and not the polls, the groups. Sorry, the groups. So wait. The rooms. Okay, guys, so what do you guys agree as a group? If it's a dimer, tetramer, monomer?
We thought it was a tetramer.
A tetramer, okay. And why is a tetramer? No, you, Nicole, someone else. Someone want to explain? Yeah, it's a tetramer, but why is a tetramer? Why do you conclude that? Any volunteer?
I just assume that because if you do the math, you can see that the SDS version is a fourth of the native protein.
Can you repeat that again?
The weight or the size that they detected under SDS is a fourth of the one that they detected when they ran it with the native protein. So that's how I got to a fourth or 4 units.
Okay. And Micaela, are you there? Okay, something like that, yes. But what else? What about the molecular weight? We have 92,000 Daltons or 92 kilodaltons and 23 kilodaltons. No one? Samantha, are you there? I want to hear the other voices.
Yes.
Hi, Samantha. Tell me, are you agree that it's a tetramer?
Yeah, I do agree.
Okay. Why do you agree?
Based on the molecular weight.
Okay. What about the molecular weight?
It would be like 1/4. Or
it will be what?
It would be like 4 times.
Okay, yeah, 4 times. Yeah, correct. Yeah, you're in the right direction. Yeah, it's tetramer, meaning it's for in this particular protein. Obviously, we now we know, right? If we add SCS with the nature of the protein, it's not going to be. It will disrupt the secondary structure, so the protein are not going to be the 92 kilodaltons, but instead it will be 23. In that specific case, it's running a lower one single band. That means that is 4 subunits, right? That are 23 daltons each one, which make the 92. So that's why it's a tetramer, right? What about the second part? Sol, are you there? Are you agree with the tetramer? Are we clear?
I have a question. Did you teach us anything about tetramers? Just making sure.
Tetramer means 4.
Okay, so I'm supposed to know it from like monomer, dimer, then tetramer, and stuff like that.
But if it's just a definition, but if you look at the chapter 4, one of the first slides, I believe it's a, dimer, tetramer, etc. And tetramer is just a word; it means 4.
Okay, because I immediately canceled it out because I don't remember ever hearing it, but that makes sense.
Okay, but it
Professor, I was looking at it as back to the question.
Yeah, I'm just going to show you this because
Okay.
I explained this. So, for example, you will have a different pattern when you have a native and you have SES because the effect that it has in the protein, right? If the proteins will be different, some of the proteins will be heterodimer. This dimer means 2. That means it's just 2 subunits, right? It can be the same or it can be homodimer, meaning that are either identical or not identical. So the point is that it's going to change the pattern in the gel. And when you have a ZS, it will run differently. In that case, because they're giving you the clue of the molecular weight, you can assume, well, you will see that is 23, the other is 93. So therefore, 23 4 times will give you the 92.
So sorry for the example we just did. It would have 4 bands. Would the SDS have 4 bands?
This?
Yeah.
No, we have 1 band. You see, both observe 1 band, 1 in each shell. One will be at 23 and the other in 92. So just 1 single band. Okay. For this specific example. So let's see. Nicole, are you there?
Yeah, I'm here.
Okay, so what do you think?
About the bands, where the bands would be?
Are you agree that it's a tetramer, dimer, monomer?
Yeah, I agree. It's a tetramer because the point of the SDS is to denature the protein, so it'll unfold it. So if there's more than 1 subunit of the protein, then the band will be lower because the weight of each subunit will be less than the overall protein. So, like Stephanie was saying, when you divide 92,000 by 23,000, it gives you 4. So there must be 4 subunits of the protein. of equal size.
Exactly. So pretend this is the gel one. Your partner, if you're in the lab run a gel, is have one band here and the other. It's the same protein, but one has the SDS and the other doesn't have the SDS. Okay. Next time, if you have questions about the word, not the concept. Here we're talking about concept, right? What happened with the SDS? But if you say, I don't know, I don't even know what is a tetramer, can you tell? You should ask me, okay? Because those are just dimer means D2, tetra means 4, mono 1, okay? So if those are things about the wording, and also about the concept, but if it's that hold you to answer, you have to ask me, okay?
Professor?
Yes.
So is it essentially the difference between where the band lands is because the SDS is making the protein more negative? And then the protein that's under SDS is going to travel further down the column because it's under SDS. And then and the difference between where they fall is what essentially helps you solve how many units up the polypeptide.
Yeah, the only thing, when you add SDS, you will change the pattern on the gel. It's not a column. We're not talking about column. It's a gel.
Okay.
Yes, in this case, because it in nature, right? It disrupt the protein is not native anymore. It will change how the protein, if it's in this case 4 subunits, then we will have the 4 subunits separate. But turns out that the molecular weight for the subunits are the same, which is 23,000 Daltons. That's why you see one single band. Okay. If you have a protein that has different subunits with different molecular weights, then perhaps you will see 2. But in this case, is 1. Ooh, let me mute.
Ezra, can you mute yourself, please?
Yeah, there's someone. Okay. What about the second part? The second part, what do you guys agree?
I think it's B, Professor.
I think otherwise. I think it's C.
Yeah, I think it's
John, why do you think it's D?
B. Let me go back to, because B said folded the protein into smaller, more compact protein, allowing it to funnel through the more easily. So,
but the purpose of the SDS is to the protein to make it smaller. What are the other things? What are you guys say to Joan?
What choice did she pick?
She picked B.
Oh, wait, what did B say again? It's not up anymore.
Oh, it's not again. Okay, wait. It says folded the protein into a smaller, more compact protein.
Doesn't it do the opposite?
If it did that, wouldn't it have like a bigger mass or similar mass in the second test instead of 23?
So what is the purpose of the SDS?
To denature the protein, so to break it down.
To denature the protein.
Correct. That's the purpose of the protein. The purpose of the SDS to the protein, right? To give a uniform negative charge, and it will denature the protein. Therefore, I thought I was sharing my screen, but let me go back. You see now, right? So the answer was basically denature the protein, which is made of equal size monomer for this particular exercise, okay? So, yeah, basically it's It was tetramer, and then the answer was the nature of the protein, okay? Any questions? Joanne, are you clear now?
Yes, Professor, thank you.
Okay. Okay. So I'm gonna talk about one more technique and then we're gonna stop here. We're gonna continue on Monday with the other techniques. I know it's a lot of techniques and I really encourage you to go and sit down and make your notes, right? What are the techniques we're looking for? What are the principles? What are the, all of that. Otherwise, it's gonna be overwhelming. After this, we're gonna study proteins, hemoglobin, and then we're gonna talk about enzymes, and then we're gonna talk about cell signaling, and all those 4 are gonna be in your exam too. So if you don't start, reviewing the materials with time, is gonna be overwhelming because it's a lot of information. And I know many of you haven't done any of this, so it's very hard. That's why I like to put the videos and the illustration, because if you haven't been in the lab looking, it's very hard, and I understand. So yeah, there was one question. Someone is raising the hand.
Yeah, that's me.
Yeah.
Is there a reverse process, like reverse to SDS, that will clump all the proteins together?
To reverse the nature?
Yeah, I'm just asking. I'm wondering if there's like a process.
Remember one of the Nobel Prize that we discussed in the previous. Actually, that will be one of your assignments to tell me if that's possible or not. Okay, who's this?
I don't know. Goodbye.
But no, who's asking me? Okay, I cannot see faces, so.
Khadija.
Okay, so what if we do this? What if next class you tell us if that's possible to reverse, right, the SDS when we add it, if it's possible. Okay. And if you can go back to the chapter before I explained some of the experiments. It was a Nobel Prize, and it has to do with the folding and if it's possible to go back or not. Okay. Okay, I think it's time. It's 7:45, so I will stop here. We're gonna start with that question and continue with the techniques. Okay. So have a good night, guys.
Thanks, Professor.
Good night, Professor.
Good night, Professor.
Bye bye.